Hello, everyone, and welcome to another Terra training module. I'm Frank Ambrosio, a bioinformatics scientist with the Agin Genomics, and in this video, we will be going over the process of running the Titan Clear Labs workflow for genomic characterization on Terra.bio. Before running the workflow, one must have some data to work with. If you have not yet set up Terra integration with your Clear Labs portal, please reach out to us at support@terrapublichealth.zendesk.com, and we will assist you in setting up this integration. Now that we've confirmed we have some data to use, let's navigate to the Workflows tab and get started. So within our workspace, if we navigate to the Workflows tab, you should see the Titan Clear Labs workflow available. We will have pre-populated this into most users' workspaces. However, if you do not see the Titan Clear Labs workflow present in your Workflows tab, please see our video on importing a workflow into Terra.bio. If you do see the Titan Clear Labs workflow, you're in luck. Click on the workflow, and you will be brought to the Titan Clear Labs workflow page, where you will be presented with some options. The first option is the version of the workflow you would like to run. We recommend running the latest version of the workflow unless you have a particular reason for sticking with an earlier version. The next option is the root entity. In this case, we will be using the sample data table. This is the name of the data table into which data is imported from the Clear Labs workflow. So we are using that as our example. Next, you must select your inputs. Here, we can select all of our inputs by clicking this checkbox or using this dropdown. If we wanted to filter by run ID, we could do so by selecting one of the run IDs and using the search function in the top right over here. This will allow us to select just Clear Labs 2 as the run. However, for today's example, we're going to be selecting all the data, so we can X out of that. Select all of the boxes, click OK. The next thing we must do is specify our inputs. We will be using the this dot notation to indicate we are using our root entity, this, and then the column from the data table of our root entity that includes the data we want to input for this variable. So for the FASTQ files, we want to use the reads variable. So this is the column header that contains our FASTQ files. The next input is a workspace element. So this is not in our Terra data table. This is a workspace element that is accessible from any of the workflows within our workspace. If you have not already uploaded a primer bed file for your workspace element, please see our video on uploading workspace elements to Terra.bio. So in this case, we'll be using the Arctic v3 primers. However, we do have other options available. If we wanted to use the Midnight primers, we could do so. However, this data was generated using the Arctic v3 primers, so let's go ahead and use that. And then we have to indicate the sample names, which we'll do by indicating our root entity with the underscore ID on the end of it. This is just how you select the sample name column from your Terra data table. If you remember in cell 1A, that's where you define your root entity. So this is the column header for your sample name column. All right, so now that we have our inputs defined, we go to the outputs tag, we click we click use defaults, and then we click save. And we are ready to run our analysis. So let's click run. Let's say Clear Labs training run, and we can go ahead and click launch. And we will be brought to the job history page where we can see the status of each of our samples individually. And we'll see this status move from queued to submitted to running to complete, hopefully. If you have any questions about how to monitor the status of a run, we do have a video on that, so please check it out as well. Otherwise, reach out to us at support@terrapublichealth.zendesk.com, and we will include a link to that support email in the description of this video. So thank you very much for watching this Terra training module, and we hope to see you again soon. Thanks.